Hello, my name is Christoph Emmerich, and I would like to give you an overview about the GOTIT project, which we conducted a couple of years ago and which focused on developing a new set of guidelines how to validate and assess a newly identified molecular drug target. But let me first introduce the team behind the project. We were basically three partner organizations coming together within the GOTIT consortium, with Ulrich Diernagel, Lorena Martinez-Gamboa, Pascal Schendel, and Mark Bonin-Andresen from the Charité in Berlin, with Mike Parnham and Martina Hoffmann from the former Fraunhofer Group Translational Medicine and Pharmacology in Frankfurt, and Anton Besbalov, Björn Gerlach, and myself from PASP in Heidelberg. And I should mention that the project was funded by the Ministry for Education and Research in Germany. The way the overall funding scheme was structured is shown here. On the one hand, 11 so-called validation projects were selected from different biomedical fields, mainly from an academic environment. They all had identified a new potential drug target as a basis for their project, and it was their task now to experimentally validate this target. Importantly, all validation projects included also a mentor who was a drug discovery expert with industry experience. And our team was then selected to develop new target validation or target assessment guidelines in parallel to these validation projects. So we had regular rounds of discussions, meetings, and online conferences with the validation teams to get their feedback and to basically make sure that the guidelines were fit for purpose, but also sufficiently flexible to cover all of these different indications so that they could indeed be implemented in an academic environment. We also discussed the guidelines during the development phase with multiple experts from industry, funders, technology transfer offices, societies, to understand their needs, get their input, and to benefit from their experience. So if you look at the drug development value chain, the GOTIT guidelines only cover a small section of this chain at the very beginning, after a new drug target candidate has been identified. But validating a target properly and understanding its value and commercial potential is important to lay a solid basis or foundation for the whole drug R&D program and for successfully reaching potential goals like obtaining funding, partnering with industry, creating new spin-off companies, or starting first-in-man clinical trials. So overall, the GOTIT guidelines were developed to help scientists with their resource management. Which activities do I need to prioritize? Help scientists to increase the value of a project, but also to help increase the confidence and the quality of evidence that we have indeed selected the right target, which is of course important for decision making regarding the next steps towards reaching the specified goals. The main idea behind the GOTIT guidelines is based on the critical path concept. Once we have identified a new target, what are the critical activities we need to conduct to reach a certain goal? Again, it could be a spin-off formation, partnering with industry, or starting phase 1 clinical trials. And how do we structure these activities in the most meaningful and resource-protecting way? This is indeed an important aspect, as it could also mean that we have to potentially terminate a project, and I will come to this in a minute. So overall, the new guidelines had to be meaningful in a sense that they can provide some real guidance for scientists starting a new translational project, but also flexible enough to cover all the different projects and indications, and any proposed guideline recommendations have to be feasible so that they can be implemented in an academic environment, as mentioned before. The GOTIT guidelines also need to play a role when it comes to changing the mindset of academic scientists. As scientists, we need to decide when a quality of evidence level is actually reached to enable certain decisions and to move on to the next activity. Or in other words, we must decide on the point at which further gains in knowledge no longer justify any additional investment of resources, because any new findings do not bring us any closer to the goal. Defining go, no-go decisions and running killer experiments are certainly concepts not all academic scientists are familiar with. But they are important to either increase the confidence in a target or to potentially invalidate or terminate a project and thereby saving valuable resources. The same concept or idea is shown here in this two-dimensional approach. So we have the scientific academic vector on the one side and the drug development vector on the other side. And we now need to understand and analyze what the critical steps and questions are which we need to answer to reach our goal. Of course, some basic academic research questions are important. For example, to understand whether a modulation of the target is indeed causing a disease, or whether any target changes are actually only a consequence of the disease and only correlate with the disease phenotype. But there has to be a focus on relevant activities towards developing a new drug or treatment for patients, as otherwise we invest more and more resources and time, but do not get any closer to this goal. And we hope that this focus and prioritization of activities is provided by the GOTIT guidelines. To follow the critical path concept, we have, as a first step, defined 5 different so-called assessment blocks, which cover different and critical aspects of target validation and the target assessment process. As you can see here, these assessment blocks deal with defining the link between target and disease, target-specific safety issues, the technical feasibility of a project, the innovation potential of a project, and any aspects important when working on non-human targets. So overall, these assessment blocks help to define the main activities and tasks of most target validation projects and target assessment procedures. So to identify and prioritize assessment blocks, we have created a set of so-called critical path guiding questions, which help scientists to get a better understanding of which are the critical tasks to be performed in order to reach their specific goals. So these critical path questions really help to select relevant assessment blocks and also help to allocate resources in the most meaningful way. The next step is to understand and to identify any relevant tasks and activities for each selected and relevant assessment block. Here we have generated a set of what we called experimental approach questions, which help scientists to define specific tasks and activities for their project. So that means that the proposed workflow when using the GOTIT guidelines looks like this. The critical path questions help to select relevant assessment blocks, and the experimental approach questions then help to identify experiments, tasks, and activities for each of the selected assessment blocks. This approach also allows to define milestones and go/no-go decision points for each assessment block, which should be considered before the next assessment block can then be addressed. Very importantly, we have also included a set of data quality questions, and these help to ensure that critical studies or decision-enabling studies have a sufficiently high level of internal and ideally external validity. And these quality-related questions need to be considered before any experiments are started, as most quality aspects also impact or influence the design of a study. So we have included guiding questions to raise awareness for different quality measures like blinding, randomization, or the need for performing a proper power calculation. Importantly, the guidelines don't say you have to implement certain quality measures, but we want to stimulate scientists to make an active decision to either implement selected measures because it is needed to generate study results which are as unbiased as possible, or to be able to justify why certain quality requirements have not been implemented. Other data quality related areas and aspects which are covered by GOTIT are the quality of tools and reagents like cell line authentication and antibody specificity, and measures to increase the external validity of outcomes and to increase the quality of evidence, for example, by using more than one model system or readout. The guidelines also focus on critical requirements related to reporting and documentation practices, as this is an area where most academic research labs most likely could improve their internal procedures when it comes to good documentation practices. And good documentation also becomes more and more critical the closer a translation project gets to interacting with regulatory authorities or starting a collaboration with pharma companies, for example. We have published the GOTIC guidelines last year in Nature Reviews Drug Discovery. So if you want to get some more details about the guidelines and the different assessment blocks and guiding questions, please have a look here at this article, which should be open access and freely available. We have also created what we called the GOTIT Target Validation Resource Center, which you will find under this link gotit.app. And it contains useful resources related to biomedical target validation. For example, a publication library and several tools like an educational platform for students and early career scientists to get familiar with the topic of target validation and to highlight some potential issues and roadblocks when it comes to properly validating a new target. And we have created the so-called critical path generator, which I will shortly introduce on the next slide. The critical path generator is basically an automated online version of the GOTIT guidelines, which guides scientists through the GOTIT workflow. After completing onboarding questions to provide some background information and to define the goals of the project, scientists are asked to fill out the critical path questions, and based on these, they will be presented with a customized set of experimental approach questions relevant for their project. And after completing these, the critical path generator will provide a summary of how the critical path could look like, as shown here under point 3. For each assessment block, there's also the opportunity to add free text to provide any details for the design of the next project-specific experiment, which the critical path generator, of course, cannot know. And all the information can then be exported and saved and shared with colleagues or collaboration partners. So the main focus group for the GOTIT guidelines are certainly academic scientists who are moving from basic to translational or applied research. And as discussed in the beginning, the GOTIT guidelines want to help them to focus on any aspects related to drug R&D which are not related to basic research, to prioritize the activities and to avoid wasting any resources, to increase the attractiveness of the project for future partners, either investors, funders, or pharma collaborators, to help define criteria for either pursuing or actually terminating a project before too much time and money is wasted, and to ultimately increase the trust between partners, as the guidelines can hopefully help to align on certain data quality criteria and de-risking aspects of a project, for example. But the guidelines could also be potentially very useful for funders, biotech investors, or industry partners, because they can be used to review a project proposal before the project starts to see whether a meaningful critical path is actually visible or not for a certain project. Can any gaps along the critical path be identified? Have resources been allocated properly depending on the specified project goals? Can any potential roadblocks on the critical path be addressed? The guidelines can support academic industry interaction by aligning quality criteria and expectations for applied preclinical research. But the guidelines can also be used to kind of measure the success or performance of a project or funding scheme and to see which milestones along the critical path have been achieved and whether any tasks and activities have been structured properly. I would finally like to thank all the different PIs and mentors from the validation projects for their input and discussions, and all the experts from different academic research organizations, technology transfer offices, funders, pharma companies, for their contributions and suggestions how to improve the GOTIT guidelines. And again, the German Ministry for Education and Research and the project management organization VDI, VDE, IT for their support and guidance. Thank you very much.